Thanks very much, Alex. Thanks, everybody. I'll just get the clicker. Thanks, everybody, for hanging around till the last session. So I'd like to just sorry? Oh, no, sorry. So I'd like to just start by thanking the organisers for what's been such a great meeting so far, and we're still looking forward to the field trips. And also to acknowledge my various supporters and funders that have allowed me to come here. So for the next 15 minutes or so, I'm going to talk to you about an idea that's been knocking around for a little while now, but I believe is gathering increasing evidence. So in modern ecosystems, skeletons are hugely diverse in terms of their function, their morphology, their mineralogy, and the ecologies related to them. And as we've heard in many excellent talks over the last couple of days, in the latest Ediacaran and started in the Lower Cambrian, we also see a huge diversity of animal skeletons. Now, many of these still remain enigmatic, but increasingly they're being placed as members of modern skeletonizing groups. And also the small shelly fossils, they show us major changes in ecology through this interval. And in fact, the origin of biomineralization is an integral part of this complex series of factors that brought about these major changes through the Ediacaran-Cambrian transition that we're all here to talk about. So for the remainder of my talk, I'm going to investigate a little bit more the origin of animal skeletons, and in particular the relationship between changes in animal genomes and the evolution of animal skeletons. So in 2011, Irwin and colleagues provided this compilation of the toolkit of messenger RNAs across animals compared with microRNAs that are involved in regulation. So if we compare when during the evolution of animals these different molecules were acquired, so if we have a look at the messenger RNAs, were acquired mostly prior to the split between bilaterians and cnidarians. And then subsequently we see predominantly a story of loss, whereas in if we look at the microRNAs involved in regulation, very few are evolved in the deepest animal branches. Most, in fact, evolved in the bilaterian stem, and then also lots of specific elements to do with the individual phyla. So there's also evidence for this inherited toolkit in the skeletons of modern organisms. So that's exemplified by this study by Jackson and colleagues. So they compared the skeleton building gene sets of two mollusks. So this is the abalone and the pearl oyster. And they also compared that to the skeletogenesis in the early embryo of the purple sea urchin. So what they found was different mollusk groups independently evolved the ability to deposit nacre. So they discovered very few similarities in the transcriptomes of these two mollusks. So at some point since they diverged around about the Cambrian, Precambrian boundary, they've independently acquired this ability. But crucially, the genes involved in biomineralization that they do share are also shared with the distantly related sea urchin. So this is a deuterostome, a completely different part of the animal's tree. So they suggested that ancestral genes were independently exapted into a biomineralization role in these disparate organisms. So this has been termed the biomineralization toolkit hypothesis, whereby a common set of ancestral genes independently exapted into building skeletons in different lineages at the same time. So we see a similar pattern if we compare across deuterostomes. So here we have an analysis of the skeletogenic gene regulatory network of sea urchins. You see if you compare that with those in their deuterostome cousins, the vertebrates, there is an extensive common biomineralization toolkit. So even though we're comparing phosphatic and calcareous independently acquired skeletons, they have this common toolkit. So do we see any direct evidence for this in the fossil record of the earliest animal skeletons? So to try and address this question, I've used the first appearance data for animal phyla and classes from this compilation, as well as the mineralogy of different Ediacaran-Cambrian and Ordovician skeletal animals, published here in 2012, along with a few other descriptions published subsequently. So what we've got here is a suite of biomineralizing taxa, and each of these bars represent a time bin for their first appearance. So these aren't the absolute ranges, these are just the when they first appear in the fossil record. So these are stage level, or down in the lower Cambrian they're local stage level. And they're color-coded based on the mineralogy. So we've got aragonite, calcitic, mixed or uncertain calcareous forms, phosphatic forms, and a little bit of silica in there as well. So similar to other compilations, we can see successive waves of first appearances of different skeletonizing taxa with a whole range of different mineralogies going through from the latest Ediacaran up into around about the sort of middle to late Ordovician. So if we then plot this data onto a consensus tree of animal phylogeny, we can see that these different mineral types are intercalated with each other. And we don't see any evidence for a single origin and then subsequent modifications of that skeleton. So I think this is evidence that we've got multiple origins of the skeleton in different animal groups. So just to take that a little bit further, let's zoom in on one part of the tree. So these are the conchiferan mollusks with shells today. And we can find examples of all of the 5 main conchiferan mollusk groups in the lower Cambrian, either as stem or possibly even crown representatives. And we can see they have a huge range of different mineralizations. So there's aragonitic and calcitic forms, and also mixed calcareous forms. But if we have a look in more detail of the textures that these different skeletons display, there's even more variability. So the very earliest representative of conchiferan mollusks have various lamellifibrillar fabrics, like this Aldanella here, and then these give way to lath shape, and then subsequently foliated fabrics in different mineralization, in different mineral systems in some cases, and independently in different groups. And then ultimately we see the origin of mother of pearl in multiple times later on in the Ordovician. So this led Vendrasco and colleagues to build this model for multiple independent origins of molluscan skeletons. So we start off with they inherit from an ancestor this thick organic scaffold, and then in the early Cambrian we get mineralization of the inner layer with these lamellifibrillar fabrics, and then the acquisition of foliated and prismatic, foliated and prismatic textures, and ultimately the acquisition of nacre. So the crucial thing that I want you to take away is that this represents increasing control over biomineralization in separate mollusk groups in parallel. So what about the other Lophotrochozoans? So here Luo and colleagues, they compared the chitin synthase and bone morphogenic protein signaling, as well as different mineralogies in mollusks and brachiopods, and came up with 3 different hypotheses for the evolution of the common skeleton of these 2 groups. Now hopefully I've already convinced you that these 2 hypotheses can't be correct, because the common ancestor of brachiopods and mollusks didn't have a skeleton. These are independently acquired. And so that leaves us with this hypothesis, whereby both these groups inherited a chitinous scaffold from which they subsequently went on to build their skeleton. So let's have a closer look at the early evolution of brachiopods. So there's increasing evidence that all of the major brachiopod clades can trace their origins to organophosphatic microfossils in Cambrian stages 2 and 3. So even the different calcitic brachiopods likely independently acquired the ability to secrete calcite, because they derive from phosphatic skeletons. And also, given the fact that we don't see any evidence of bona fide stem brachiopods with a phosphatic skeleton, it may well be the case that we have independently acquired phosphatic skeletons in each of these different brachiopod groups, and even in possibly in the phoronids, which may nest inside the brachiopods, have phosphatic ancestors down in the Cambrian. So again, we're seeing multiple origins of the skeleton within a clade. And again, if we look at the microtextures that these potential fossils that are placed in these stem positions possess, we see similar microtextures in fossils placed in different parts of the tree. And also we see a great deal of variation within, so this is a Sonogenia sclerite, so we see variation, these are both Sonogenia. So we've got variation within sclerites of the same taxa, and also but also fundamentally similar structures where you have this predominantly organic sclerite that's been phosphatized. And also there's the potential for lophophorate in the Lady Ediacaran, which also possesses these simple foliated textures in the form of Nama calathus. So that's another potential. So to summarize this first part of my talk then, so despite having independent origins for the brachiopod and the mollusk skeleton, they share a whole range of different features. So for example, the shell pore systems, the organic rich shell, mantle and periostracum, complex layered shell later on in their evolution, and also similar shell microstructures. There's also evidence to suggest that bryozoans share similar microstructures as well, and it may well be the case, although remains to be tested, that other lophotrochozoans that in the modern are largely unmineralized, but we have mineralized examples down in the Cambrian, may also exhibit similar microstructures. So overall then, the model that I'm gathering evidence to test is that we have this inherited organic scaffold, which independently in different lineages you see a laminar microstructure initially under loose control, and then subsequently more derived entrenched microstructures later on in each of these lineages. So this story is not restricted to the lophotrochozoans. So if we return to this study by Luo and colleagues, so they demonstrated that the genes involved in biomineralization in vertebrates, in mollusks, and in brachiopods are different. Okay, so they're not directly shared. However, if you look at which genes these mollusks are using to build their skeletons, many of them you can find in human and brachiopod genomes, but they have a diverse range of different functions, cellular and metabolic processes. So this suggests that this suite of 30 genes, for example, that are found in all of these genomes, they were independently co-opted for a role in biomineralization in the mollusks. And it's very likely that the vertebrate skeleton also evolved by the calcification of an organic scaffold. And this has gained even more evidence recently by the identification of a collagenous skeleton in the Cephalochordae amphioxus. So this is the collagen just here. So the generally accepted model for the evolution of vertebrate mineralization is you start off with a collagenous scaffold that's initial mineralization utilized using these spark genes, and then through a series of whole genome or partial genome duplications, you get increasing complexity of skeletal tissues independently acquired in different lineages, and also increasing complexity in fibrillar collagens. And the fossil record of early vertebrates largely agrees with this. So we see down here we've got the plesiomorphic vertebrate skeleton, and we see independent modifications of that skeleton in each of the major skeletonizing vertebrate groups. And if we examine the evolution of the conodont skeleton, so these are known to be analogous to vertebrate teeth. Again, we see the earliest examples are largely organic with mineralization under loose control, so they likely evolved by calcification of a cyclostome-like feeding apparatus. And then as we move up towards euconodonts, we get increasing complexity in the basal body, and then ultimately get the evolution of this highly organized euconodont apomorphy, the crown tissue. And as we're going to hear, it's nearly finished. As we're going to hear much more from the next speaker, there's even evidence that in the Ediacaran we've got the utilization of organic scaffolds, and then the evolution of skeleton under loose control. But again, we're going to hear much more than that from Rachel. And just one final twist in the tail. There's also evidence to suggest that horizontal gene transfer has played a role in the co-opting of these genes in a range of different bilaterians, and also in increasing evidence in sponges as well. So to summarize then, there's evidence both from fossils and from molecules for multiple independent origins of skeletons, even within individual phyla or even classes. But they've all inherited this organic scaffold and a genetic framework from their common ancestor. And we see this repeating pattern of increasing complexity and control in the evolution of a range of different skeletonizing groups. So thank you very much for your attention.